In this practice session, we will use the Mega package for creating phylogenetic trees. You can download it through the following link. Choose the latest version or an older one. Then fill in your details and click on Download. We will use version X for this practice. Let's think about the following scenario. We have a Canadian patient that got measles after returning from a vacation in Nice, France. We want to identify the source of his infection. Was he infected while he was in France? As you know, the measles virus is highly contagious and is spread by coughing and sneezing via close personal contact or direct contact with secretions. In order to better understand the source of the infection for our patient, we'll construct a phylogenetic tree for the measles virus. We have sequences of the envelope glycoprotein from the virus surface, which is called hemagglutinin H. They were collected from patients in several locations around the world, including Canadian patients, French patients, and South Asian patients. We will build a phylogenetic tree from 5 Canadian patients and 3 patients from Nice in France. Our goal is to know if our Canadian patients was infected by a Canadian or French measles strain. In order to gain confidence in the results of the tree construction and to be able to identify its root, it is advised to add an outgroup sequence that should come out as an outlier in the final tree. Therefore, we added a sequence of a Chinese isolate of the virus. The first stage is to create a multiple sequence alignment. You can either do it within Mega or import a multiple sequence alignment from a different program such as the clastel-O that we previously discussed, as long as they are saved in Mega format with the .align extension. Here we will align the sequences within Mega. Open the Mega program, click on Align, choose Edit Build Alignment, choose Create a New Alignment and click OK. If you want to import a multiple sequence alignment, choose the option Retrieve a sequence from a file. At the next window, choose DNA for building a DNA sequence alignment. Now copy and paste your sequences to the Open Alignment window. Do not forget to give informative names for each sequence, as these names will be presented at the final phylogenetic tree. So you should edit the description line at the FASTA format sequences by writing the relevant name directly after the bigger than sign. In our example, we added the country name at the beginning of each description line. The Mega program offers 2 commonly used alignment algorithm options, MASL and clastel-W. Click on the alignment at the main menu and choose Align by clastel-W codons. This option is used to align via clastel-W the coding sequence data in the current selection by first translating all codons to amino acids, performing the alignment, and finally replacing the amino acids with the original codons. Click OK to choose all sequences. At the Open window, use default parameters values and click OK. Click Yes to confirm using the standard genetic code. Now you can scroll on the alignment to see the conserved columns marked with stars and the added gaps. Next, we need to save the alignment in Mega format. Click on Data, Export Alignment, Mega format. Give the alignment a name and click Save. At the new window, type this name and click OK. Finally, click on Yes for ensuring that these are protein coding nucleotide sequences. Go back to the main program window and click on Phylogeny. This program offers a variety of phylogenetic algorithms like neighbor joining, maximum parsimony, UPGMA, and more. Here we will demonstrate the use of neighbor joining, but as always, we recommend repeating the construction with another algorithm and comparing the results for consistency. Choose the neighbor joining option. Then, choose the alignment file that you saved. In the parameter window, change the test of phylogeny to Bootstrap method. Write 1000 under number of Bootstrap replication. Bootstrapping here means that the tree is constructed many times by sampling a partial subset of sequences, and the results are tested for internal consistency. You can use the Help button to get more explanation on each parameter. For example, under the Gaps or Missing Data Treatment parameter, you may choose to remove all sites containing alignment gaps and missing information before the calculation begins by choosing the Complete Deletion option. Alternatively, you may choose to retain all such sites initially, excluding them as necessary in the pairwise distance estimation by choosing Pairwise Deletion option. Or you may use partial deletion site coverage as a percentages. We will use the default values recommended. Click on Compute. First, reassuringly, we can see that indeed the Chinese gene was clustered separately from all the other ones, suggesting it originated from a different strain. All the Canadian viruses were clustered under the same ancestor, meaning they were probably originated from the same strain, whereas all patients from France were clustered together under another ancestor. We can clearly see that the gene from the Canadian tourist is included in the French group, suggesting that he was infected while he traveling in France. The number next to each node represents the bootstrapping percent number. A high value means that there is a strong evidence that the sequences to the right of the node cluster together as it showed up in most of the bootstrap replicates. We can present just bootstrapping values that are higher than 70 by clicking on View, Options, Branches, then clicking on Statistical or Frequency, and writing 70 under Hide Values Lower Than, and clicking OK. You can also present the tree in different formats. Click on View, using Tree Branch Style, and then choose one of the suggested styles. The phylogenetic tree can be presented in two ways. Phylogram. The branch lengths are proportional to the amount of changes along the branch. Cladogram. The branches are of equal length, we can see a common ancestry, but it doesn't indicate the amount of evolutionary time separating taxa. You can switch between the phylogram and cladogram formats by clicking on View Topology only. Let's color all the French and Canadian patient virus branches in a different color in order to make them distinct from the rest. First, click on their common node representing their common ancestor. Click on the hammer sign appearing in the left toolbar. Change the branch line to orange and click OK. Note there are a lot of other design options you can use. We can also add the branch length. The branch lengths usually represent the evolutionary distances between two consecutive nodes. The length of the branch is proportional to the number of changes. The distance between two species is the sum of the length of all branches connecting them. Click on View, then Show Hide. Finally, choose Branch Length. In order to remove the branch length, click on Branch Length once more. And now you know how to create a phylogenetic tree.